Good afternoon, everybody. This is chapter 10. I guess I should say good morning or just hi, since I don't know when you're watching this. We're in the muscular system, and chapter 10 and 11 are all about muscle. There are three types of muscle that you've already learned about from chapter 4 when we talked about the different muscle types. But this chapter is going to focus mainly on skeletal muscle, which you see on this individual who is playing tennis. So the three types of muscle tissue that we have, skeletal muscle on the top, which we'll talk about some characteristics that differ between skeletal, and then we have smooth muscle in the middle and cardiac muscle on the bottom. And so in this chapter, though, we're going to focus on skeletal muscle. We will talk a little bit about cardiac and smooth muscle and how each of these muscles are similar and different, okay? So one thing that all of these muscles have in common, though, is their basic functions. So all muscle has certain characteristics, no matter what type of muscle they are. And these characteristics include responsiveness. So what that means is they are excitable. They will respond to certain types of chemical signals. They have a plasma membrane that is highly excitable. And when something excites that plasma membrane, they will then conduct that excitability, there's the word I want to use, along the plasma membrane and down into the muscle itself, into the muscle's cytoplasm. When muscles get stimulated, they will contract, and what contraction means is that they're going to shorten, and then they are extensible, which means they can also stretch out. So once they contract, they will relax, and a lot of times they will stretch further than their relaxed state. Elasticity means that they're going to go back to their resting length once they have contracted and extended beyond that normal length. So skeletal muscle has a specific organization, and skeletal muscle fibers are long in length, and I'm just going to go back to show you, right here. If you look up here, this is one muscle fiber, here's another muscle fiber, here's another muscle fiber, here's another muscle fiber. So they're lengthy. They have all these nuclei along the edge. They're multinucleate. So muscle fiber, or an entire muscle, is composed of anywhere from, just a few muscle fibers to hundreds and hundreds of muscle fibers. These muscle fibers are then bundled into fascicles. A fascicle is a bundle, then a group of muscle fibers. And so those larger muscles, like the leg muscles that we have, these muscles are going to have multiple fascicles that make up the muscle. And the entire muscle is going to have a special wrapping around it called the epimysium. The wrapping around that separates each fascicle of muscle fibers from each other is the perimysium. And then surrounding each individual muscle fiber is an endomysium. The epimysium and perimysium are composed of dense irregular connective tissue, and they bring in, so they separate muscle fibers, they bring in nutrients to the muscles, but surrounding each of the muscle fibers individually is an areolar connective tissue, and that helps to insulate each muscle fiber from all of its neighboring muscle fibers. And that's really important because each muscle fiber can be individually stimulated. And so you don't stimulate, when you have a muscle contraction, not every single muscle fiber in the muscle is contracting at the same time. Different muscle fibers within that muscle are contracting. This is one way that we are able to continuously stimulate muscle for long lengths of time. Think about standing for 8 hours. You could not continuously stand for 8 hours if the same muscle fibers were contracting and relaxing. Eventually they would get tired and you would end up having to sit or lay down. So this keeps those muscles moving by stimulating different muscle fibers at different times. The epimysium that surrounds each muscle runs into the tendon. The tendon is an attachment. It's composed of dense regular connective tissue. It attaches your muscle to bone. This is what allows us to move our bones. A flattened tendon is called an aponeurosis. You have flattened tendons on the top of your head and around your wrist and your ankles. So what are muscles composed of? There were initially, just like everything, we have the mesoderm is what gave rise to our muscle. And some of the mesoderm specialized into forming myoblasts. Myoblasts form skeletal muscle. And so this is kind of my drawing of what myoblasts kind of looked like. They were tiny little stem cells, and when they came together, they, instead of fusing together and forming an individual cell, that's exactly what they did, but they retained their nucleus. And so you ended up having multiple nuclei within one cell. And so there's your skeletal muscle fiber in the end. Muscle fibers have a special plasma membrane called the sarcolemma. Anytime you talk about anything, you're dealing with muscle. And so the sarcolemma is the plasma membrane of a muscle fiber, and it surrounds the entire muscle fiber, plasma membrane, so muscle cell. The epimysium and the sarcolemma run together then as well. An individual muscle fiber is composed of multiple proteins called myofibers or myofibrils. These myofibrils form what we call a sarcomere. They form structures called sarcomeres. And one single muscle fiber can have multiple sarcomeres. So I'm showing you that here too, just to show you. Invaginations of the sarcolemma are called T-tubules. And so along the sarcolemma, at specific regions, the sarcolemma invaginated or pushed in on itself and pushed into the middle of that muscle fiber. And those structures have a special, so those structures are called T-tubules, and attached to those T-tubules is the Sarcoplasmic Reticulum. So this like orangish structure along the side here, and this orangish structure here, that's Sarcoplasmic Reticulum. The Sarcoplasmic Reticulum of skeletal muscle is a specialized smooth endoplasmic reticulum that contains calcium. So it has a major function of harboring calcium ions for the muscle fiber. And I already mentioned this, but myofibrils are what make up your muscle fiber, and proteins make up those myofibrils. And the functional unit of a muscle fiber then is a sarcomere. So each muscle fiber can have multiple sarcomeres that run from one Z disc to another Z disc. So this entire structure is a sarcomere. And there's special names for each region of the sarcomere. The Z discs are the boundaries. Then we have these dark purple structures here. Anywhere that we have these thick filaments located, that's known as the A band. It's where thick filaments are located. Anywhere that there's only thick filaments and no thin filaments, so I should mention these green lines are the thin filaments. The area where there's only thick filaments is called the H zone. Area where there's only thin filaments is called the I band. So what are the thick filaments and the thin filaments? Thick filaments are called myosin, and the thin filaments, there are multiple types, and I'll show you here. Those little bead-looking structures, those are actin proteins. The string-like structure here, that yellow one, that's tropomyosin, and then do you see these other little bead structures like there? You can see it up here, and you can see it right there. That's troponin. So there's 3 types of thin filaments that we talk about and 1 thick filament that we talk about. The myosin and actin are called contractile proteins, whereas troponin and tropomyosin are called regulatory proteins. So skeletal muscle does not contract without nervous innervation. Or, I mean, you can artificially stimulate skeletal muscle as well. But in a living individual, you know, when we think about moving our muscles, only nervous innervation can stimulate muscles to contract. So if you have nerve damage, then you're not going to be able to use your muscles properly. And if you remind me, I can show you what that looks like because I do have nerve damage in my hand, and I'm able to show you what nerve damage does to the muscle, or what nerve damage does and how it affects the muscle. That's probably a better response to what I just said. So we have somatic motor neurons that innervate our muscles. So these neurons, or they innervate our skeletal muscle. So somatic means skeletal system. And so these somatic, or motor system, I should say, these neurons then innervate our skeletal muscle, and they form a junction between the neuron head and the muscle fiber itself, okay? They don't actually touch, so this is called the neuromuscular junction, just so you know. And each nerve, so a nerve is composed of multiple neurons, and each neuron has multiple axons, or not, no, that's not true. Multiple end bulbs, that's what I want to say, axon terminals. And each axon terminal is going to innervate only one muscle fiber. So only one muscle fiber is going to have one axon terminal innervating it, but one neuron can innervate multiple fibers. And so one nerve can stimulate multiple muscle fiber groups, if that makes sense to you. But any given muscle fiber will only have one single neuron stimulating it from the same group. So the synapse that you see is a gap between the neuron end bulb and the muscle sarcolemma. So that little gap is a region. Neurons don't actually touch their target cell. Instead, they release chemicals called neurotransmitters into that gap, and those chemicals stimulate the next cell. And so for muscles, skeletal muscle, the neurotransmitter is acetylcholine, and it's found in these little tiny vesicles. And stimulated, when an action potential stimulates the axon end bulb, then that acetylcholine from the vesicle is going to be released into that synapse. And the acetylcholine will bind to receptors on the muscle fiber. And when two acetylcholine bind, then sodium-gated channels open up and sodium rushes in. So I mentioned the T-tubules before, and the reason I did was because those are invaginations of the plasma membrane or the sarcolemma. When an action potential stimulates the skeletal muscle fiber, the sarcolemma, that excitation moves along the plasma membrane and down those T-tubules to get deep into that muscle fiber. And when it does that, because we have the sarcoplasmic reticulum attached directly to those T-tubules, making that little triad structure, then the sarcoplasmic reticulum also gets stimulated. And the sarcoplasmic reticulum contains bound calcium ions, which we need for a muscle contraction. So when a muscle contraction is going to occur, calcium has to be released into the muscle fiber. Calcium moves in and it will bind to the troponin. Troponin and tropomyosin are attached to actin. The tropomyosin covers the binding site on actin. And so when troponin binds to the calcium, troponin and tropomyosin move off of that binding site, and that allows the binding site for myosin to attach, or the binding site for myosin to attach to the actin molecule. So if you look closely at the myosin head here, it has two different binding sites. It has a binding site for the actin. It also has a binding site for ATP. And so the myosin molecule is going to bind to the actin when calcium is present, but if calcium was absent, then the tropomyosin would be covering those little binding sites on the actin molecule. So I want to go through the entire process in order. I'm going to stop this now, and then I'm going to start the next lecture, and we're going to go over every single step of the muscle contraction. I'm going to start at the neuromuscular junction again, but I'm going to walk you through it. We've gotten through the majority of the contraction already, but I'm going to start over in our next video because it's really important. I want you to understand what's going on, okay? So I'm going to stop here, and then we'll start our next video, well, in part 2 of chapter 10. Bye.